Okay, the topic we'll be dealing about is immunity. Immunity is the resistance of the host towards the different types of microorganism or its product. Resistant towards the microorganism is the body is trying to defend itself from the different types of microorganism as well as its products. So in case of certain organism, it is the only organism that causes the infection while multiplying. Whereas in case of certain organism, these organism can produce the disease by producing certain types of toxic products which are called as toxins. So it is the resistance of the host to protect itself from the different types of microorganism or the infections by the microorganism or from its products, that is the toxins that are produced by the microorganism. So our body has developed different types of mechanism to resist from these type of organism that causes the infections in our body. So this is called as an immune response towards the foreign substances. Foreign substances or the foreign antigens are nothing but the organism which are pathogenic in nature and has the ability to cause the infection. So these foreign antigens can be any. It can be a bacteria, it can be a virus, it can be a protozoa or any organism that has the ability to cause an infection in the human body. So it need not be any organism, but it can also be certain types of chemicals that are like pathogenic or that causes a deleterious effect in our body. So immunity is resistance towards the microorganisms and its product, and it is called as an immune response towards a foreign pathogen, and the foreign pathogen can be any type of organism that can initiate an infection and cause the infection in the human body. Now the classification of immunity, the immunity can be classified basically into two types. One is called the first line of the defense and the second one is called as the second line of defense. Only after crossing these first line and second line, the organism is able to cause an infection in the human body. So it is not easy for an organism to cause an infection in the human body. It has to cross these barriers, only then it will be able to cause the infection in the human body. So the first line of defense that comes directly to the microorganism or prevents the entry of the microorganism is called as the innate immunity, also called as a non-specific immunity. It is so called because it is not specific towards a particular organism. That is, it does not protect our body only against one type of organism. It has a broad activity towards different types of organisms. That's why it is called as a non-specific mechanism or non-specific immunity, also called as innate or native immunity, that is usually already present in the human body. And the other type of immunity is called as the acquired immunity or adaptive immunity. Acquired or adaptive immunity is the one that we get during the course of infection. That is called as the acquired. We acquire it during the course of infection or during the course when we interact with a particular type of organism. So the innate immunity is said to be non-specific, whereas acquired immunity is said to be specific towards a specific type of organism. That is, once we get an infection towards a particular type of organism, next time we will not get that infection because we already have immunity towards that particular organism. That's why it is called as specific immunity. Acquired immunity, also called as adaptive immunity, or also called as specific immunity. So here again we can classify innate or non-specific or native immunity into non-specific and specific type. Here we have species immunity, we have racial immunity, and we have individual immunity, because all individuals are not same. So we differ from each other from person wise as well as from the area wise as well as the environmental wise. So depending upon all these conditions, you can say that there is a species immunity, there is a racial immunity, and there is also an individual immunity. Whereas in acquired immunity, you can classify it into two major types. One is called as the active immunity and passive immunity, which I will be dealing later. Now first, coming to the innate immunity or native immunity. It is the genetic constituent of a particular human being. It is a genetic or constituent makeup of a human being. That is, he is made up like that to prevent himself from the different types of diseases. It is this genetic component determines whether he has to be susceptible or resistant towards a particular organism. And these type of components are already present in our body to fight against the organism or the incoming foreign antigen. And it is said to be non-specific because it has an elaborate activity. It does not protect us from a particular type of infection, but different types of infection caused by different types of organisms. Hence it is called as non-specific immunity. And this is be classified as species immunity, racial immunity, and we have individual immunity. Now coming to the species immunity, it is a group of people, or a species of people who are resistant towards a particular type of infection. Take for example, we all humans are unsusceptible to plant pathogens. So that is called as a species immunity. We don't get the diseases that the plant gets. The organism that causes the diseases in plant cannot affect us. So that is called as a species immunity. So we all are resistant towards a particular infection that is caused in other group of species. So that is called as a species immunity. Next we have a racial immunity. Within a species, races also differ in their immune responses or immunity towards different type of infection. Take for example Africans. Africans are more resistant to malaria than the Asians. So here the races are different and hence the immunity towards that particular organism is also different. So within species, races also shows the difference in their immunity. And ultimately coming to individual. So one individual is different from the other individual. One can be susceptible to a particular organism whereas the other can be resistant towards the same type of organism. This can be very well explained in homozygous twins and heterozygous twins. Homozygous twins shows resistance and susceptibility similar to the same type of infection, but whereas heterozygous twins usually shows different types of response towards a particular organism. One can be resistant, the same organism towards which the other person can be susceptible. So it is within the races, each individual can differ from the immune responses towards a particular type of organism. So innate immunity is non-specific and can be classified as species immunity, racial immunity and individual immunity. Apart from that, we also have herd immunity. Herd immunity means in a particular area, a group of person, if they are resistant towards a particular organism, then it cannot lead to epidemic. If the group of person in a herd are susceptible to a particular infection, then it can lead to epidemic. So it is depending upon the herd, whether it is resistant or susceptible, it can lead to epidemics or endemic or pandemic diseases. Now next coming to the factors that usually are influencing the innate immunity. Now there are different factors that influences the different persons on their immune responses. So first I will deal with the age. So it is, we can say very well say that very young people and very old people are susceptible to different types of infections. They are not resistant. So whereas in the adults you can see they are resistant towards different types of infection. The reason behind the young and the old being more susceptible to infection is because of the immune apparatus. The immune apparatus in the very young people is not very well developed, whereas the immune apparatus in the very old has deteriorated. That is the reason that they are more susceptible to different types of infections. Now the organism that has let us go with the ages. So first I will deal with the fetus, then we'll go with the young, then adult and then with the old. So first with the fetus, the organism which has the ability to cross the placenta and cause the infection in the fetus are the rubella, herpes, toxoplasma gondii, etc. They have the ability to cross the placenta and cause the infection in the fetus, and the fetus affected with this can either die or can be born stillbirth or with different types of deformities. So the fetus cannot protect himself because he himself does not have the immune apparatus to protect from the diseases, whereas it is the mother's antibodies that usually protects the fetus from the different types of infection. So once the baby is born, during few weeks he is on the maternal antibodies that protects him from different types of diseases. Once he is developing into 3 to 4 months, the immune apparatus starts developing and protects the baby from the different types of infection. Meanwhile, if the organism is able to cross the placenta and cause the infection, then it can lead to the deleterious effects on the fetus. Now next coming to the young, it is usually the hormonal influences, you can say, that can lead to either susceptibility or resistance towards a particular infection. Young are mostly susceptible because the immune apparatus is still on the going process to develop in the young individuals, and it is the hormonal influences also that keeps on changing, leads to different types of infection in the young generation or young people. Example, tinea capitis causes Microsporium ordini during the puberty. Once the puberty sets in, there is a resistance towards this type of infection. Now coming to the adults, adults are more prone to diseases such as poliomyelitis due to hypersensitivity reactions, and moreover there are only few diseases that can affect the adults compared to the young and the old. Next factor is the hormonal influences that usually makes a person resistant or susceptible towards the diseases. So here we have endocrine disorders such as diabetes mellitus, we have adrenal dysfunctions, we have hypothyroidism that can lead to the person susceptible to different types of diseases. Take for example, in a diabetic mellitus patient, he is more prone to staphylococcus sepsis, where the carbohydrate content is more in a diabetic patient. So this takes a favorable advantages for the staphylococcus sepsis to cause sepsis in the diabetic mellitus patient. And then we have corticosteroids, which has both beneficial and harmful role in the human body. The harmful role that it gives to the human is it depresses the host resistant towards different types of infection. That means presence of corticosteroids make the person more liable to get affected with different types of diseases. Apart from that, the beneficial role that is given by corticosteroids is that it protects the person from the bacterial products such as toxins. So depending upon the corticosteroids presence, it can also make a person resistant or susceptible to a particular infection. And the increase in the steroid levels during the pregnancy also makes the pregnant woman more susceptible to different types of infections. And then we have a stress that is usually brings about the different hormonal imbalances in our body, leading to different types of infection. So hormonal influence also have a predominant effect on the person's immune system. Next factor is the nutrition. There is a complex relationship between the malnutrition and immunity. It is very well said that a person who is on a well-balanced diet is more resistant towards different types of infection, whereas the person who is malnourished can be affected by different types of diseases. But there are certain diseases which can be expressed only in an healthy individual. For example, malarial fever cannot be expressed in the malnutritional people, whereas the person, if he is on a good diet, then the fever can be expressed in the malarial patient. So there is a complex relationship between the nutrition and the immunity. Next is we have barriers of innate immune system. So barriers of innate immune systems are divided into 4 that prevents the entry of the organism and prevents the diseases in the human body. So one is the anatomical barriers, physiological barriers, third is the cellular immune factor, and fourth is the inflammatory response. So first I will deal with the anatomical barriers. The barriers that prevents the entry of the organism into the host to cause the infection are called the anatomical barriers. The best example of this is skin and the mucous membrane. Skin, as such, we know it's a layer that usually gives us a shape and protects our body against a wide variety of components that are trying to gain an entry into the human body. And apart from that, the pH of the skin being acidic in nature due to the sweat also will not allow the entry of the organism or makes the skin a hostile membrane to not allow the growth of the organism. So it acts as a mechanical barrier to prevent the entry of the organism, and due to its dryness also the organism cannot survive because they require certain moisture for their survivability. And the activity of the sebum underlying the skin also contains certain types of antibacterial agents that will kill the organism when they are trying to multiply inside the skin. One example of this is that overcomes the factors of skin and the sebum is a propionibacterium acne that has the ability to metabolize sebum and cause the infection called as acne. Otherwise, no organism will have the ability to multiply on the skin and cause the infection or gain an entry until unless there is any wound or a pricking that inoculates the organism inside the skin. And then below the skin we have the different areas or the organs of the body such as respiratory organ, we have genitourinary tract, we have gastrointestinal tract. All these tracts are usually covered by a mucous membrane, which is a thin membrane and secretes mucous substance which is called as mucus, and this mucus will try to trap the organism and will collect the organism and transfer it to the stomach, where they are killed by the acidity or the pH of the hydrochloric acid, and the acidity of the gastric juices usually kills the organism. So in the mouth it is a saliva. If the organism is trying to gain an entry through the mouth, it is a saliva that contains the lysozyme and different enzyme that will kill the organism. And if the organism is trying to gain an entry via the nostrils or the nose, the organisms are first collected through the nostril orifice, and they are taken up by the mucus again to the stomach, where they are killed. Apart from that, sneezing, coughing also act as a protective role to eliminate the organisms. And then we have conjunctiva, that is a lining of the eyes that prevents the entry of the organism due to lacrimal secretion. And apart from that, tears are also produced. Now this tears contains an enzyme lysozyme that will kill the organism and will not allow the entry of the organism. Next factor is the urine. The flushing action of the urine also will prevent the exit of the organism from the body, and the acidity of the urine is such that it will not allow the growth of the organism. Apart from that, the reproductive organs, that is the semen produced from the reproductive organ of the male, contains spermine and the zinc that will have a bacteriolytic activity. And in the female, the vaginal pH is low, as a result of which the microorganism will not be able to multiply and cause the infection. So these are the anatomical barriers that will prevent the entry of the organism. Apart from that, it is the help of the normal flora that will not allow the pathogenic organism to establish on the human host and cause the infection. So this concept is called as microbial antagonism or antagonism of indigenous flora. Indigenous flora are nothing but the normal flora or the normal microorganism that is present in the human body in harmony with the human body. They do not cause any type of infection. They are called as commensals. But these organisms becomes pathogenic when the immune system or the immune status of a person comes down. Otherwise, they are usually as beneficial to the human being as we are benefiting the organism. So this organism will compete with the pathogenic microorganism for the food and the place, and as a result of which will not allow the growth of the pathogenic organism or attachment of the pathogenic organism on the human host. So these are all the anatomical barriers that prevent the entry of the organism. Next, coming to the physiological barriers. So we, as discussed, there are four barriers: anatomical barriers, physiological barriers, we have phagocytosis, and ultimately the inflammatory response that will eliminate the organism. So now next, coming to the physiological barriers. The physiological barriers will include all kind of chemical substances that are produced by the human body to prevent the infection. The first is fever. So whenever we have an infection, there is a sudden increase in the temperature of the body. So fever is a protective response towards a microorganism, because at high pH the microorganism will not be able to survive. And the example that can be given, the organism which is very susceptible to high temperature is the Trypanosoma pallidum. So this organism, when there is an increase in the temperature due to a change in the thermoregulatory system of the body, there is a hypothalamus, there is an increase in the temperature, and ultimately the organism gets killed due to high temperature. So it is a protective role towards the microorganism. And next is the acidic pH. The pH of the skin, the pH of the stomach contents are usually acidic in nature, which usually are harmful to the microorganism, and microorganism gets easily killed with the acidic pH. Next are the lysozyme. Lysozyme is a secretions produced by different parts of our body. They are present in the skin, they are present in the tears, they are present in the saliva, and this lysozyme usually will inhibit the organism by not forming the cell wall. So they are the one that usually acts on the cell wall of the organism. Now when they act on the cell wall of the organism will die due to the release of the content of the organism or due to the osmolysis of the organism. So ultimately it is the death of the organism due to the factor which is called as the lysozyme. And apart from that, acute phase proteins are also produced. The best example of acute phase protein that is produced is called as a C-reactive protein, and the C-reactive protein that is produced during an inflammatory response or during any type of infection will activate the complement system by alternative pathway, and it will ultimately kill the organism by formation of the holes on the microorganism. And they also act against the different types of bacterial products such as toxins. And next important physiological barrier is a bacteriocin produced by the different components or the cells of the immune system that are highly active against the bacteria, and we have leukins produced by polymorpholeukocyte that kills the microorganisms, and we have phagocytin produced by the macrophages that helps in the engulfment of the organisms, and we have beta lysins, thermostable substances that are active against the anthrax bacilli and the typhoid bacilli, and we have spermine which is present in this sperms, the male reproductive organ that kills the organism. And next is the complement proteins. Complement proteins are nothing but the serum proteins which are present in the serum of the human individual, and these complement proteins usually are activated by three pathways, ultimately forming a complex which is called as a membrane attack complex on the organism, and the organism gets killed due to its activity. And then we have interferons, which are antiviral substances. So before that we have seen all the antibacterial substances, and it is the antiferons that is the antiviral substances. They are naturally produced proteins that are active against the virus. So they create an antiviral state so that the virus cannot initiate the replication process in the cells that have been attacked with the viruses. So the interferons are antiviral substances. So all these are the components that are included in the physiological barriers. Now next, coming to the cellular factors involved in innate immunity are the phagocytes. Phagocytes are nothing but the macrophages, which are the cells of the immune system, and it was first discovered by Elie Mishnikov, who stated about the concept of phagocytosis, where the foreign antigens are usually engulfed by the macrophages and they are usually killed inside the phagocytes by different contents that are present in the phagocytes. Examples are the macrophages, and here the organisms are usually taken up by a process which is called as a phagocytosis, and they are enclosed in a vesicle which is called as a phagosome. Now the phagosome fuses with the lysosome that is present inside the macrophages to form a complex which is called as phagolysosome. Now the lytic component that are present in the lysosomes are treated upon on the organism, or they kill the organism that is present in the phagosome. Ultimately, the degraded particles are excluded from the phagocyte by a process which is called as exocytosis. So the phagocytes are the major important component of the immune system that straddles between both innate as well as acquired immunity. They activate both innate immunity as well as the acquired immunity. It is with the problem with the capsulated organisms. The capsulated organisms are highly virulent in nature because they can escape the phagocytosis. Otherwise, any foreign particle can be easily engulfed with the help of phagocytes. And next we have the other type of cells, that is a natural killer cells. They are non-specific cells that usually are active against the viral infection and the tumor cells. That is, they detect the viral infected cells in our body and the tumor cell, and they will try to eliminate these cells from our body. So they are called as natural killer cells. Next next type of barrier is called as the inflammatory response. The most important type of barrier which helps to eliminate the organism from the body by a series of reaction that is taking place after there is a tissue injury or the irritation or a wound that is caused by pricking or the inoculation of the organism into the human host. So this is called as inflammatory response. There are 4 cardinal signs of inflammatory response, that is called as rubor, calor, tumor and dolor. Rubor is redness or erythema, calor is increase in the temperature, tumor is edema, and dolor is the pain. So when there is an inflammatory response, you can see all the 4 cardinal signs that are brought about during an inflammatory response. So when there is a tissue injury or there is an entry of the organism through the wounds, the arterial system first constricts and then dilates, due to which more amount of blood flows in that particular area where there is a tissue injury. That leads to rubor, that is the erythema, redness in that particular area. And then we have a tumor because apart from the RBCs, we also have the phagocytes and the other cells of the immune system coming to that area to eliminate the organism. Hence there is an increase in the size of that particular area which is called as edema. And next we have increase in the temperature due to the flushing action of the blood towards that particular area wherever there is an injury. There is an increase in the temperature that leads to calorie, increase in the calorie. That's why it is called as a calor. And finally, the pain receptors produces certain types of components which are called as bradykinin that stimulates the pain in that particular area. So we have all the 4 cardinal signs that is taking place during an inflammatory response, that is rubor, calor, tumor and dolor. So ultimately, when this inflammatory response is being taken up, there is a fibrin clot that is formed, and this fibrin clot will prevent the spread of the infection. It acts as a wall off between the lesion and the healthy area. It will not allow the organism to make an entry or gain an entry and spread the infection. So this is all taking up during an inflammatory response to prevent the organism from gaining an entry into the human body and cause the infection. And this is about the 4 important components of innate immunity. Innate immunity, non-specific immunity or native immunity. It is because of the genetic constituent of the host, and it has 4 barriers. One is the anatomical barriers, physiological barriers. We have phagocytosis and we have the inflammatory response. Now coming to the next type of immunity, which is called as acquired immunity, also called as adaptive immunity. Now this is so called because we acquire it during the lifetime. It is only after an interaction with the antigen or the organism we develop this kind of immunity. That's why it is called as acquired immunity. And here in this acquired immunity, you have 2 important types of component. One is called as a recognition and next is the response. So the acquired immunity is so specific that it is able to recognize the organism and it gives a response. The response is of 2 types. One is called as the effector response. The other one is called as the memory response. Effector response is the initial response towards the organism. So immediate response that we give towards a particular organism is called as the effector response, and memory response is the one that is on subsequent infection with the same type of organism. The memory cells keep circulating in our body, and during the next time of infection, these memory cells will go and interact and eliminate the organism. So the main component of acquired immunity is to recognize the organism, that is whether it is a foreign organism or a self thing, and then it has to respond towards the foreign organism, not towards the cell thing. Okay, so the response is 2 types, effector response and memory response. So this acquired immunity can be again classified into 2 types, active immunity and passive immunity, which is further classified as natural active immunity and artificial active immunity. And then we have passive immunity again classified as natural passive immunity and artificial passive immunity. So first I will deal with the active immunity. Active immunity is developed only when there is an antigenic stimulus. As I told you, there should be an interaction between the antigen or the foreign particle to develop this kind of immunity. So that is called as active immunity against the antigenic stimulus. And it is also called as adaptive immunity because we adapt it during our lifetime. It is not already present as we have seen in the innate immunity. It is we adapt it during our lifetime when we interact with the antigen. It is called as adaptive immunity. And the components of the active immunity or the acquired immunity are the production of antibodies and the other cells. So the most important component that goes and interacts with the antigen are the antibodies. So during this acquired immunity, there is a latent phase and a negative phase too. Latent phase is because it takes some time for the immune system to develop. So it is giving some time. So it is like you can say opposite of innate immunity, which is already present and can interact with the antigen. Whereas in case of acquired immunity, it is usually produced only when there is an antigenic stimulus, and it takes some time for the immune system to develop to go and interact with that particular antigen. Now there is a negative phase, as I told you. It's because the cells which are already present in our body will interact with the antigen that has come. So there is a negative phase in that. Whatever the cells are there, they have been used up. So it takes some time for the new cells to be formed to go and interact with that particular antigen. And apart from that, we also have a memory response. The response is towards a subsequent infection. So initially when there is an development of immunity, 2 cells are formed. One is the effector cells and the memory cells. So effector cell immediately interact with the antigen, whereas the memory response is still present in our body for subsequent infection, and it is more rapid when compared to the effector response because there is no need of the immune system to get ready at that time. Already cells are there, they can go and interact with the antigen. So here the active immunity can be divided as natural active immunity and artificial active immunity. So here in the natural active immunity, there is a clinical or inapparent infection from the microorganism. The best example is the measles. Once we contract that measles, in the next part of life, we will not get that particular infection because we already have immunity towards that particular measles. So it is long lasting and it varies with the type of pathogen. And here we have bacterial infections which are less permanent in nature. Now coming to the artificial active immunity, the best example is the vaccines that are produced from the organism. Vaccinization or immunization is nothing but making the antigenic preparation. So antigen is taken and it is removed with the antigenic property, but it has the ability to induce the immune response. So the vaccines that are produced from these kind of antigens are used for immunization purpose. So these vaccines can either be a live vaccine or a killed vaccine, and the process of preventing person from the diseases by using vaccines is called as immunization. And we have different types of vaccines for different types of organisms, such as bacterial vaccines are there, viral vaccines are there. Bacterial vaccines, take for example, they are live vaccine and killed vaccine. So live vaccine, in the live vaccine we remove the antigenic character, but we will not kill the organism. But in case of killed vaccines, the organism is killed, but it has the ability to induce the immune response. Take for example the cholera vaccine is a killed vaccine. Similarly, we have viral vaccines, which is a live vaccine given as a oral polio vaccines, and then we also have the vaccine for the viruses which are called as SAC vaccines, which are killed viral vaccines against the polio diseases. And these live vaccines need not be given repeatedly, whereas killed vaccines, because they show only temporary effect, they have to given again and again in the form of booster doses. And then the enhancement of this vaccines or virulence of the organism is called as exaltation, whereas the decrease in the virulence is called as it is called as attenuation. So attenuated organism can be made to boost an immune system by adding an adjuvant. So we have got different types of adjuvants that can combine with the vaccine to enhance the immunogenicity of a particular disease. Next is the passive immunity. The passive immunity is also called as a ready-made form of immunity, because here there is no antigenic stimulus, no organism is involved, but the treatment is given directly towards a particular antigen, where the person who is not able to produce that immune competent cell towards that particular antigen. In that case, direct antibodies are given in a ready-made form. So no active role of recipient over here, because the person is not able to show the immune status to protect himself against that particular disease, and there is no antigenic stimulus, and here we do not have immunological memory. It is only transient period of time. Okay, only for a particular period of time, only till that particular infection is there, we can use this passive immunity. Give the live prepared antibody forms to the person to protect him from the diseases, where the immune status is not very well developed in the person, and it is less effective when compared to that of the active immunity, and it is immediate and instant immunity. Now coming to the classification of passive immunity, it can be classified as natural passive immunity and artificial passive immunity. The best example of natural passive immunity is the immunity provided to the fetus by the mother. So mother to the baby. So here we have different antibodies of the mother that have the ability to cross the placenta and protect the fetus from the different types of infections. And the antibodies through the colostrum, that is the first formed milk, can have different types of antibodies. When given to the child will be protected against the different types of infections. So it is a maternal antibodies that protects the fetus, and it's the best example of natural passive immunity. So it protects from all common diseases till the baby develops his own immune system, and it is an active immunization of mother pregnancy. Next is artificial passive immunity. Here we give the antibodies that are prepared in the laboratory, okay, from the horse serum or from the experimental animal or from the human who voluntarily come up to give the antibodies. So antibodies are preformed in the laboratory and given to the patients who are suffering from various diseases such as snake bites. Immediately they have to be given with certain types of antitoxins, right? So antibodies acts as antitoxin to prevent from the snake venom. And here we have hyper immune sera of animals that act as the preformed antibodies or human antibodies. We have convalescent sera, we have pooled human gamma globulin that acts as an example of preformed antibodies. So it is immediate and temporary because it will not give any time of long lasting protection. No memory response is seen in this. And you now also have a combined immunization with the active and passive immunity called as adaptive immunity. So this can also be, the work is being carried out in this particular area to prevent a person from the different infection, both activating the natural immunity, that is the active immunity, as well as the passive immunity. So this is all about the immunity of a particular person who protects himself from the different types of diseases.